This conference will now be recorded.
Okay, great. Chalo. So let us also take the quick recap of what we have learned in the last session. We have started with that DNA is the basic, uh, DNA is the unit of heredity, right? We have started with that DNA is the unit of heredity. And we learned that how the DNA is made up of, it is made up of nucleotides, right? It's made up of nucleotides. Is my screen visible to everyone? Yes, is it visible? Is it visible to everyone? Okay, chalo. Nice. So, nucleotides we learned, right? Nucleotides is the basic unit of DNA, and we also learned the difference between nucleotide and nucleoside. So, today we have to learn that how this DNA is compactly packed into the cells and what is chromatin and their parts we are going to talk about in today's session, right? So here we know that the size of the DNA molecule present in eukaryotic organism or prokaryotic organism comparatively is very large, right? The size of DNA molecule is comparatively very large with the cell size. What I am saying, the size of the DNA molecule is comparatively very large to respect to the cell size. Okay, even we talk about the cells or we talk about the viral particles, right, which are the connecting link between the living and non-living. So the even though it is viral particle, right, viral particle is consist of only a protein layer, right? That is called as coat. So within this coat, there is genetic material present. Either it could be DNA or RNA. So the length of this DNA or RNA, or it is in case of cell inside the nucleus or in case of a prokaryotic cell within the cytoplasm. So the length of all these DNAs, the length of all this DNA is comparatively larger than the cell size. So now how this DNA is placed in such a small space, right? So with the help of tertiary packaging of DNA, how it is possible? Because of tertiary packaging of DNA, packaging of DNA into chromosomes. So what are chromosomes? So now we get to know that chromosomes, they are nothing but compactly packed DNA, right? Which is also called as tertiary structure of DNA. So chromosome is nothing but this tertiary structure of DNA. So let us now clear with the concept what is mean by the primary structure. What is mean by the primary structure, secondary structure and tertiary structure? Is there any problem with my screen? Is my writing is visible, students? You can answer me by unmuting also, right? No, no, it's not visible. Okay, my writing is not visible. Okay, just give me a second. Okay, is my screen visible now? And whatever there in the screen, is it visible?
Yes, ma'am.
Yes, okay. Chalo. So what we were discussing that how this large DNA is packed into the cell. So with the help of its tertiary structure. Tertiary structure means compactly packed structure. So now we want to clear what is mean by primary structure, secondary structure and tertiary structure in case of any molecule. Okay, so normally the primary structure of any molecule means the monomer units join to each other in a straight chain. Monomer units join to each other in a straight chain. This straight chain structure is called as primary structure. This straight chain structure is called as primary structure. So in case of DNA, we say nucleotides attached to each other with the help of which bond? Can you tell me which bond? Nucleotides are attached to each other with the help of which bond? Yes, unmute and tell me. Yesterday itself we learned.
Phosphodiester.
Very good. With the yes, it's a type of covalent bond, but more specifically, we call it as phosphodiester bond because all the molecules, okay, proteins, all the macromolecules, protein, DNA, lipids, they are made by joining the monomers, and to join all the monomers to give rise a primary structure, we usually make covalent bond. Okay, so we have different name to this covalent bond. So here we use phosphodiester bond, right? Phosphodiester bond. So the nucleotides attached to each other with the help of phosphodiester bond giving rise to a chain. This is the primary structure. Okay, and when these two chains of the DNA, two strands of the DNA, they are attached to each other by the hydrogen bonding within the nitrogen bases, and these two strands, they are intercoiled on a imaginary central axis, right? They are intercoiled on a imaginary central axis. This is called as secondary structure of DNA. This is called as what? Secondary structure of DNA, where the two strands, they are bonded to each other with the help of hydrogen bonds, and again these two strands, they are bonded with each other to give a helical structure, right? To give a helical structure. So this is nothing but the secondary structure. Now when this helical DNA is packed with the help of proteins, yes, yesterday we discussed, so this structure is called as tertiary structure. So primary structure mostly consists of covalent bond. Secondary and tertiary structures involves non-covalent bonds, that is called as weak interactions, right? To to form the secondary or tertiary structure of any molecule. If it is protein or DNA, we require the weak interaction, or we call them as non-covalent interactions. Okay, this much clear? So now see, if I say, so breaking of this weak interaction or non-covalent interaction is very easy. Okay, these bonds, they are reversible. They can be immediately broken with some condition, with some environmental condition. They can be rebonded again. Okay, so this is called as, whenever we are breaking the secondary bonds or tertiary bonds or secondary or tertiary structure in any molecule and making it back to primary structure. Okay, or gaining its primary structure again. See, we have, whenever we have primary structure, from that we get three three-dimensional structure, right? That is called as tertiary structure. Three-dimensional structure, or we say tertiary structure, right? So these are reversible structures. Okay, let me clear the screen and we'll discuss further. So here we can see, so whenever there is primary structure, it can be converted into tertiary structure, right? Now we can convert back tertiary structure into primary structure, and this is called as denaturation. This is called as what? Denaturation. This we have to remember, denaturation. And again, after denaturation, if the primary structure is again converted into tertiary structure, that is called as renaturation. Okay, these are two terms important: denaturation and renaturation. Primary structure to secondary tertiary structure is renaturation. Tertiary structure to coming back to the primary structure is denaturation. So can we say chromosome, as we have already mentioned, chromosome, it's a tertiary structure, right? So whenever we are converting back chromosome into double stranded DNA, and from that double stranded DNA, if we are getting two separate strands of DNA, if we are converting this double stranded DNA, first it was in chromosome form, from chromosome form we have, or we can draw chromosome like this, right? Compactly packed structure, it looks like this. So chromosome to a double stranded DNA, double stranded DNA to the single two different strand we are converting. This complete process is denaturation. Okay, if again we are converting this two single strands to into double strand, this process is renaturation. What this process is? Renaturation. Okay, but you know what? So breaking down weak interaction is easier, but breaking down covalent bonds is difficult. Okay, it requires very heavy treatments. So if we want to break the primary structure of DNA, if we want to break the primary structure of DNA, so what we will get? We will get individual nucleotides, right? If we break the primary structure of DNA, we will get the individual nucleotide. So this is called as hydro, or this can be achieved by the hydrolysis. Okay, and this can be achieved by which process? Hydrolysis. And this is called as degradation of DNA. What it is called as? Degradation of DNA. So once we do degradation of DNA, reobtaining DNA is very difficult. Then we have to synthesize the DNA, the primary structure again. But if we do denaturation, we can easily renaturate it, or we can easily again make it functional. So see, most of the molecule like DNA and proteins, they are functional. DNA and protein, they are functional in their three-dimensional structure, mostly proteins. Okay, so if we denature them, they become non-functional. So to be functional, the formation of 3D structure is important. So secondary and tertiary structure is important. So making secondary tertiary structure from primary structure is also called as folding. Is also called as what? Folding. Getting this much, students? Everyone clear?
Yes, ma'am.
Yes? Okay, great. So these are the terms which are useful. So see, whenever anybody says denatured DNA, so you should always keep in mind there are two separate strands of DNA and which can be again bonded together by the hydrogen bond in between the nitrogen bases. But if anyone saying degradation of DNA, so you should understand the individual nucleotides are being broken down. So degradation of DNA will, so we cannot obtain it further, right? Again. Chalo. So, cellular DNA. Now, let's talk about the concept of gene. Now, we will talk about the concept of gene and then we will talk about the chromatin. So, now let's start with the gene. So, we all know that DNA is the genetic material, means DNA is coding for all the required characters in all the individuals, right? DNA is coding for characters, right? Or we can say it is coding for phenotype, physical characters. All the physical characters visible are because of the presence of DNA, right? Phenotype means what? Appearance of character is called as phenotype, and their code in the form of DNA is called as genotype. These two terms, genotype and phenotype. What is phenotype? Phenotype is appearance of physical character, right? Which is coded by DNA, right? Which is coded by DNA. And what is genotype? What is genotype? The code by which physical characters are coded, right? The genetic code or the DNA code we say, right? Genotype is nothing but the DNA code by which physical characteristics are coded. Okay. So we know that all the phenotypic expression are possible because of the, yes, genotype present, right? All the physical characteristics are possible because of genotype present. So we have so much longer DNA. All the organism they have much more DNA, but actual part of DNA which is coding for this phenotype or which is coding for the, so these phenotypes they are possible because of formation of protein. These phenotypes are possible because of formation of proteins, right? So all these phenotypical characters they are coded by proteins. Actually, proteins are being synthesized in our body, which are responsible for giving us the physical character. But not complete DNA do this process, or not complete DNA is responsible for making protein. So, what are genes? So, we can say that genes are, so what are genes? Genes are segments of DNA. Genes are segments of DNA that code for polypeptide. Polypeptide means protein. है ना? Polypeptide means proteins. So that code for the polypeptide chain and RNAs. Right? Because RNA also play functional or structural role in the cells. Right? So all the functional role is performed by proteins and RNAs. So in all cells, the functional role is performed by whom? The functional role is performed by proteins and RNA. So the segments of the DNA, if we have so much longer DNA, suppose this is our DNA. So in this DNA, there are some regions. In this DNA, there are few regions which the information from this regions, so getting everyone the highlighted part we are understanding. So the whatever sequence present in this region, whatever ATGC sequence present in this region, according to that sequence, the amino acids will be attached. And now after attaching amino acid, the chain formed is polypeptide chain, right? Or we say protein chain. Now this protein are going to function. Similarly, from any one of the segment, the RNA can be formed, right? Here is also first RNA is formed, and from this RNA the protein is formed. So now here RNA is doing some functional role, or this RNA is involved in making some structure in the cell. For example, ribosome, right? Ribosome is made up of protein and RNAs, right? So protein here plays the structural role, and some protein they do functional role like mRNA. They perform the function of transferring the information. tRNA, right? tRNA also performs some function of transferring amino acid. So what I am saying, students, you are understanding what is gene. So genes, they are the segments of DNA that code for either protein chains or RNAs, right? So this is the modern definition for gene, right? This is the modern definition for gene. So what are gene? Genes are the functional parts of the DNA which codes for polypeptide and RNA. Or we can say a gene is all the DNA that encodes the primary sequence of some final gene product, right? So can I write this? Have you noted all the things? Got the definition? What is gene? See, we are going to see how this definition is derived. Gene. Okay, first there was some different definitions for the gene. Got this much, students? Yes? Only Ganga got. What about others? You all can unmute and respond me. No problem. Tushar, Suchitra, Sonali, Satyam.
Yes, ma'am.
Okay, great. Chalo. So see, so now we know that this is the definition of gene, right? And we also understood what is now exactly gene. But for the very first time, this term gene was coined, or the gene was described by, what are actually genes is described by two scientists, Biddle and Tatum. What are the name of scientists? Biddle and Tatum. So they have described the gene in 1940. Okay, yes. So very first time they have tried to describe the gene, what is actually gene, because they get to know that all the DNA we have, that is not functional, that is not coding for gene. But this doesn't mean that we are carrying unnecessarily DNA. Okay, there are, see, now we get to know that there are some coding functions or coding part, and which doesn't code, this is called as non-coding part. But both coding and non-coding part are equally important for all the cells. That's why they are carrying that, right? है ना? We living organism, we are very intelligent, right? We do not carry anything without our, क्या हमारे मतलब के बिना हम कुछ नहीं करते, right? So it doesn't mean that we are unnecessarily carrying the non-coding DNA. Non-coding DNA also has a very important role, as they help in mutation, avoiding mutation and evolution, right? They help in mutation and evolution. Okay, and coding part is making a cell functional, right? So very first time they coded, they described that Biddle and Totem. What is gene? So gene is a segment of genetic material that determines or codes for one enzyme. What they have just stated? What is gene? Gene is a part of DNA which codes for one enzyme. And the hypothesis come, they have given a hypothesis means a consideration they have given, right? And a hypothesis means a suppose, right? So they have given the hypothesis called the one gene one enzyme hypothesis. ओके, ये नाम सुन के हमको लगता है कि बाबा ये क्या है, बट इट इज नथिंग. दे हैव स्टेटेड दैट the functional part of the DNA is gene, right? And one gene is going to code for only one enzyme, always. That they have stated. But this is not the case, right? The one gene, one enzyme hypothesis they have given. But this hypothesis was right to some extent, but not applicable for all the genes, right? But they have started, at least they have given this one gene one enzyme hypothesis. Many a time the question being asked, who given the one gene one enzyme hypothesis, what this hypothesis state? So this hypothesis state that one segment of the DNA, one functional segment of the DNA will code for one enzyme. So what are enzymes? So can anybody tell me the definition of enzyme? Yes, what is enzyme?
Enzymes are the proteins.
Very good. Enzymes are the protein. Then why we are calling them as enzyme? Why not proteins?
All
That
proteins
is
are
right.
not enzymes.
Oh, yes, right. See, this is very correct that enzymes are proteins, right? Or we say all enzymes are proteins, right? But all proteins are not enzymes. This statement, this assertion is right. But why we are calling some proteins by different name called enzyme? वो कुछ तो अलग करते होंगे ना, इसलिए उनको हमने different name दिया है, right?
Ma'am, the functions as the biological catalyst for biochemical
Very
processes.
right. Okay, very good. Who has given this answer? May I know the name? Yes?
Tushar.
Tushar. Okay, Tushar. Right, very right. So, what are enzymes? Enzymes are nothing but the proteins which acts as biological catalyst. Acts as biological catalyst. Now, what is catalyst? See, students, whatever concepts will require for studying all the biology or all the life science. Okay, whenever the concept is coming, we are taking in detail at that point. Okay, like we're discussing now enzymes and catalyst. Okay, the topic which is required for us, right? So, what is catalyst? So, catalyst is anything, right? Catalyst is anything which increases the rate of reaction, which increases the rate of reaction. It means it makes the reaction to happen fast, right? So it could be any reaction, it makes the reaction faster. That is called as catalyst. And the same role of catalyst is performed in case of biochemical reaction or the reactions which are ongoing inside the cell. So to make that reaction fast, who is functioning? Proteins are functioning, right? So we have given that proteins a name as enzyme. So enzymes, can we say now enzymes is biological catalyst? Clear? So they are going to increase the rate of reaction, they are making reaction faster. And how do they do it? By lowering the kinetic energy. Okay, how exactly the catalyst they increases the rate of reaction, or enzymes they increases the rate of reaction? This is very important. Okay, on this question can be asked, okay, how the enzyme, what is the mechanism by which catalyst or enzymes increases the rate of reaction? So by lowering the kinetic energy. Right? What is kinetic energy? Kinetic energy is the energy in the things which are in motion, right? That is called as kinetic energy. So see, lower is the requirement of the kinetic energy, faster will be the reaction, right? जितना कम एनर्जी चाहिए उतना ही फास्ट होगा रिएक्शन, राइट? अगर एनर्जी रिक्वायरमेंट इज मोर, सो इट विल टेक मोर टाइम टू गेन द एनर्जी, राइट? सो catalyst, what catalyst do they arrange the reactant in a such a way that it will require the interaction will be much more greater among the reactants, and it will require less kinetic energy, and the reaction happens fast. Clear everyone? Right? So most of the functions in our body are performed by the proteins, and many of them most of them act as enzyme. So Biddle and Totem state that all the genes they code for the enzyme, and one gene will code for one enzyme only, right? So here, from here, the one gene one enzyme hypothesis came, but later on we get to know that all the proteins formed from the genes, they are not acting as enzymes. Most of them acting as enzyme, but mo- many of them they are not acting as enzymes. So they have modified this hypothesis from the one gene one hypothesis to the one gene one polypeptide. Instead of this enzyme word, what word they have taken? Polypeptide. Okay. So now the next concept, later this concept was broadened, that one gene one polypeptide, because many genes codes for protein, that is not an enzyme, right? But later on we get to know that, okay, there are so many genes and they are forming the polypeptide chains, right? They are forming the polypeptide chains. But many of time the single polypeptide chain is not able to function. It requires the binding of one polypeptide chain with the another polypeptide chain. Binding of one polypeptide chain with another polypeptide chain. We call it as multi-subunit protein, right? Multi-subunit protein. Very best example is hemoglobin, right? So hemoglobin is a protein which consists of four polypeptide chains, right? So now this hypothesis is not going right, that one gene, one protein, or one gene, one polypeptide is right, but one gene, one protein is not going right, or one gene, one enzyme is not going right, because many of the time to become functional, a enzyme required multiple peptides, right? Multiple protein chains, and that can be coded by the different genes, right? That can be coded by the different genes. So very right hypothesis is what? One gene, one polypeptide, not even one gene, one enzyme, one gene, one protein, right? Because to be a functional protein, some proteins require more than one genes, right? So now one gene, one polypeptide, definitely. But some of the genes, they are going to form only RNA, and this RNA is going to be functional or structural part, or this RNA is going to be functional or structural role, right? So now the very right definition of the gene is what? The definition which we have studied. So modern biochemical definition for a gene is all the DNA that encodes the primary sequence of some final gene product, right? Any or they, so we can say that it is what? It is the sequence on the DNA which are going to code for some final product, right? That final product could be protein or RNA that has some functional role, right? RNA that can has a structural role or catalytic function. Very clear what is gene? I hope now you all will not forget what is gene, right? Yes? Clear everybody? Yes? Good, chalo. So now we get to know that DNA contains genes, but with genes, DNA contains some other sequences as well. There are coding and there are non-coding sequences, right? We discussed there are coding sequences and there are non-coding sequences. But you know what? Instead of this, coding means which is going to give rise RNA or proteins. Non-coding means they are not going to code, or they are non-coding means they are not going to perform any function in the cell, right? They are not even going to code any RNA protein or not any function. But despite of this coding and non-coding sequences, DNA also contains one more type of sequence called regulatory sequence. What they are called as regulatory sequence. So what is regulatory sequence? So regulatory sequence are the sequences in the DNA which provides signals that may denote the beginning or end of gene, right? So we now know there are genes, but from where exactly, from which sequence the gene starts, from which sequence the gene ends, right? So that can be identified with the help of some sequence inside the DNA that are called as regulatory sequences, or there are some sequences which influences the transcription. Okay, whether the transcription should done, we in the next upcoming topics in the molecular biology we are going to talk about all this. What is transcription, translation, replication? So there are some of the part or some of the sequence in the DNA which also influences this process. Okay, this has some effect on this process, like transcription. It also there are some sequences which influences the process of transcription of a gene, right? Or there are some sequences which functions as, or they functions as initiation point for replication or recombination. Initiation point for replication or recombination. So all the sequences, rather than the coding and non-coding sequences, all the sequences which can provide a signal, or which may denote where the gene starts, where the gene ends, or these sequences which influence on the gene, or they will regulate the transcription rate, they will regulate the start of transcription, end of transcription, or start of replication, or at which point the recombination should be done. So all these sequences, they are called as regulatory sequences, right? Because they are regulating the process of transcription, replication, and recombination, right? So some genes can be expressed in different ways, right? So now there are some genes, there is one gene only, but its expression is in different ways, so that from one gene we can get different products. From one gene we can get different products because they can be transcribed, they can be expressed into the different types. Understood this much clear? What is what is DNA, what are genes, what are regulatory sequences? Hypothesis, one gene one hypothesis, or one gene one enzyme hypothesis given by Biddle and Tottle, and that is modified to now one gene one polypeptide hypothesis. Okay, not even protein, polypeptide, right? Protein is the complete functional polypeptide is called protein. Clear? Yes. So let us move ahead. So now let us discuss about the DNA in or the chromatin in eukaryotes and the chromatin in prokaryotes. So as we talk about the chromosome, right? Now we have to talk about the chromosome. So before talking about the chromosome, we know that, see, one more thing is we have to discuss about genes is, so genes, they are going to code for protein, right? So protein is your chain of amino acid, right? Chain of amino acid. So how to decide which amino acid to be added? So that is decided by the 3 nucleotide sequence of DNA. So the information to add amino acid is present in the DNA in the combination of 3 nucleotide. Okay, 3 consecutive group of nucleotide will give us information to add one amino acid. So this group of 3 nucleotides is called as codon. It's called as was codon. So see, first what is there? First there is DNA. Now the information from the DNA is converted into mRNA. This process is called transcription. This process is called what? Transcription. Now this transcribed RNA, from this transcribed RNA, the nucleotides, they are being read into three groups. So the group of three nucleotide is called as codon. So one codon will add one amino acid. So you know what? With the help of this information from the protein we can give the actual or minimum length. We can give the actual or at least minimum length of gene in the DNA. Getting what I am trying to say? If we have protein, so what we can do, if we have protein, known protein structure, or we do know how many amino acids there in the protein, if we know the length of protein, we can give the length of DNA. How we can give? As we know that one codon codes for one amino acid, and one codon contains how many nucleotides? One codon contain 3 nucleotides. So by this we can give the length of DNA in the form of base pair. How many base pair the DNA has, we can give from the protein. For example, very simple example we will take. If a protein has 350 amino acid. Okay, let's take simple example. You can solve and tell me the answer of this. I will first take a simple example. If a protein has 20 amino acid, if a protein has 20 amino acid, can we give the length of DNA? Can we give length of DNA? See, 20 amino acid means 20 codon. Each codon has 3 nucleotides. So we can simply multiply 20 into 3, right? So 3 2s are 6, that is 60 nucleotides, or we say the DNA has 60 base pairs. What is the length of the gene? 60 base pair. Understood? Yes? Clear? See here, if this example we take, this has 1, 2, 3, 4, 5, 6, 7, 8 amino acids, right? 8 amino acids means 8 nucleotide. Each nucleotide contains, 8 codons. Each codon contains 3 nucleotides. So we can simply multiply 8 into 3. So that is 24, right? So these are 24 nucleotides. Length of this DNA is 24 base pair, actually we say base pair, right? The gene is base pair. So this is one base pair, right? This is called as base pair, and in between The bonds shown are the hydrogen bond. Clear, everybody? So can you calculate now, if a protein has 350 amino acid, what will be the length of DNA in base pairs? If a protein has 350 amino acid, what will be the length of DNA in base pair? Can you calculate?
1050 base pairs.
Very nice, very good. Who has answered this? किसने दिया
Pu-
आंसर?
Purnima.
Okay, Purnima, very good, beta. So all others understood how the answer came? So if an amino acid has 350 base pair, sorry, 350, if an protein has 350 amino acid, so what will be the length of DNA? The length of gene we say, right? Length of gene is 150 base pairs. Got it? Good. Satyam, right answer. Yes. What about others? So let's move ahead and talk about now the chromosome in eukaryotes and prokaryotes. So see, these are some examples. You can note this for your information, that these are some examples of viruses and their DNA in base pair. Okay, base pair we understood, right? This is, if it is a double stranded DNA, so this is a base pair, right? You can see this is a base pair. CG is a base pair. See, in the mRNA there are only bases, right? There is not pair, and in the DNA there are base pair. These are the base pairs, right? Okay. So if we talk about the length of the DNA and the size of cell. So you can say here are some examples of viruses like 5X179 T7 bacteriophage, lambda virus, T7 virus, or T4 virus. Okay, all of these are bacteriophages. What does mean by bacteriophages? The viruses which can infect the bacteria, right? What is bacteriophages? Viruses which infects the bacteria, which infects bacteria only, right? We know that viruses are host specific. If they are able to infect bacteria, they will not infect plants and animals. If they're infecting animals, they will not infect plant and viruses, right? So see, if we talk about the viral size, let's take the example of T7. So T7 has its genome size is 39,000, right? It's quite large, 39,936 base pair. So according to this, you know what, if we are talking about the base pairs, so the length in between two base pair is 3.4 angstrom. The length in between two base pairs, the two base pair occupies 3.4 angstrom space. Okay, so according to this, if we calculate, so it has 39,936 base pairs. So according to this, the length of viral DNA becomes 14,377 nanometers. Okay, it's quite large. And what is the size of its particle? The size of its particle is only 25 nanometers. The length of DNA is 14,377, and the size of the particle is only 25 nanometer. So this much larger DNA is kept into this smaller particle. How? By the condensation, by the compact packaging of this DNA. Understood? Yes. So here are some more examples we have about uh, these are the examples of some bacteria and yeast, fungi, plants and animals. See, first example is a bacteria, Escherichia coli. It has genome size. Everyone note down the genome size of each of these, okay? Because they are being asked in the exams. Most frequently, it is the genome size of Escherichia coli, genome size of the lambda phage. Okay, in the previous page, there was the genome size of lambda phage. It is 48,592 base pairs. Have you noted this? Note down the name of organism and its genome size. Noting, everyone? Yes? May I have some responses? Okay, so very much important is keep the genome size of lambda phage, okay, lambda virus. And next important is size of Escherichia coli, genome size of Escherichia coli. Then yeast, that is Saccharomyces cerevisiae. Then you can remember the genome size of Drosophila melanogaster and humans. Okay. Actually, the table is itself very important. If possible, remember the genome size of all these mentioned organism, because these are the prototype by which we are studying the complete class. Okay, if you want to study bacteria, we only study Escherichia coli. Okay, in unicellular eukaryotes, we have studied Saccharomyces yeast, right? These are the organisms whose complete DNA is studied. How many genes they have, what's the length of their DNA, everything, right? So if we talk about the Escherichia coli, so Escherichia coli, it's an prokaryotic organism and it has only single DNA. It has only single DNA, which is also circular in nature. Okay, we can't say it's perfect circle. Okay, it is not perfect circle, but it's a DNA with endless loop, right? There is no end, it is a looped structure. So it could be like this also, but the first point and last point is joined, right? So it's a looped structure, not perfect circle, but it's looped one, right? There is no end, so it's an endless DNA. So that's why it is called as circular. So Escherichia coli, they has single circular DNA which is double stranded. They so Escherichia coli. See, we have in the topic or in the syllabus we they have mentioned us to study the prokaryotic and eukaryotic chromosome. So now we are studying the prokaryotic chromosome with the example of Escherichia coli. So how the DNA of the Escherichia coli is the DNA of the Escherichia coli is circular and double stranded. So that we say the number of fragment present or number of DNA fragment in a cell is nothing but called as chromosomes, right? Which are going to be packed compactly. So we can say that the number of chromosome in Escherichia coli is one only, right? So its genome size is this 46 lakhs 39,675 base pair, right? So you can remember it as 4.69 into 10 raised to 5 as well, right? And the gene present in this genome is 4,435. That is functional segments. Functional segments are the segments of DNA which are functional. So this is the approximate number of the genes present in an Escherichia coli. Okay, clear? Noted? So you can note the genome size of human as well. So approximately is 3.0 into 10 raised to 9, right? It is 10 raised to 9 base pairs. So human genome is 3.3 into 10 raised to 9 base pair. What is the size of human genome? 3 into 10 raised to 9 base pairs. What is the genome size of Saccharomyces cerevisiae? We can say approximately 1.2 into 10 raised to 6 base pairs. Yes? Clear? Clear, everybody? Okay. So, with this, let's go inside the E. coli genome. So, the E. coli genome it do not only contains the gene, okay, or the size of the DNA which we learnt is called as chromosomal DNA. It's called as what? Chromosomal DNA. But inside the E. coli cell, despite of this chromosomal DNA in the cytoplasm, there is the presence of some small circular DNA. So many small circular DNA are present in the E. coli cell, and this small circular DNA with short base pair with less base pair. The base pairs are it is being mostly approximately of 2000 base pairs. Okay, the DNA it is of only 2000 base pair, which is circular, and which code for some proteins which can gives the bacteria some special advantages. So such genetic material, despite of this chromosomal material inside the cell, is called as extra chromosomal DNA. It's called as what? Extra chromosomal DNA. So despite of this nuclear or the chromosomal DNA, if the cell possessing some extra DNA is called as extra chromosomal DNA, and in case of bacteria it is some circular DNA. One or many copies of such circular DNA can be present, and they are called as plasmids. They are called as what? Plasmids. Okay. So the complete genome. So whenever we use this word genome, so genome includes the complete set of DNA in the cell or in an organism. That is chromosomal DNA. That is chromosomal DNA, or we say nuclear DNA, plus extra chromosomal DNA. This is very important. Sometimes in MCQs they are being asked what is mean by genome or genome is. So genome is the nuclear or the chromosomal DNA plus extra chromosomal DNA, right? So in case of eukaryotes, the extra chromosomal DNA is plasmid, right? Extra chromosomal DNA is what? Extra chromosomal DNA is plasmid in case of eukaryotes, right? It's plasmid. So it is also defined by CCC, covalently closed, covalently closed circular DNA. How it is circular? Mostly it is circular. So these plasmids, they are only present in bacteria, some yeast, and algae. Okay, so where you can observe the plasmids you can observe in bacteria, mostly in bacteria, some yeast and algae. Okay, so most of the time we get confused. हमको हमेशा पता होता है कि सिर्फ बैक्टीरिया में ही प्लाज्मिड होते हैं, बट ऐसा नहीं है. There are few yeast and algae, they have plasmids. Okay, understood? So these are eukaryotic organisms, right? We always consider that there is only plasmids in bacteria, but few. I'm not saying all. Few. Few yeast and algae, they also has the extra chromosomal DNA. Now let us talk about the extra chromosomal. So we are clear with this now. The extra chromosomal DNA in bacteria, that is plasmid. So these plasmids, they have the ability to self-replicate, right? This plasmid, they can replicate themselves and they can provide some extra advantageous property to the bacterial cell. For example, antibiotic resistance. So this plasmid, they contain such a gene sequences which can code a protein which help this bacteria to become resistant to antibiotic, right? Getting everybody? Yes or no? Not only antibiotic resistant, there are so many features can be imparted by the plasmid, like they can give the metal resistance, okay, high metal concentration resistance, antibiotic resistant. Everybody getting? So when we are going to talk about the techniques in the biotechnology or recombination, recombinant DNA technology. That time we are going to discuss these things in detail in recombinant DNA technology. For here it is clear है सब लोग या चले गए? Students,
Yes, ma'am, it is clear.
clear? Okay. So now let us talk about the extra chromosomal DNA in eukaryotes. Okay, so eukaryotes do possess extra chromosomal DNA. Do you know? Do we have extra chromosomal DNA? हमारे पास without the nuclear DNA, is there any extra DNA present?
Yes, ma'am,
Yes.
mitochondria.
Very good. So eukaryotes also do possess the extra chromosomal DNA that is called as mitochondrial DNA, right? Mitochondrial DNA or the chloroplast DNA, right? So in case of eukaryote, mitochondrial DNA and chloroplast DNA are the extra chromosomal DNA. So this mitochondria and chloroplast DNA, they are also self-replicating. So they are called as semi-autonomous organelles. They are called as what? Semi-autonomous organelles. So many of the proteins, they are produced by the mitochondrial chloroplast DNA, but most of the proteins which are required for the mitochondria and chloroplast to function are coded by the chromosomal DNA. So that's why they are called as what? Extra chromosomal, semi-autonomous organelles, right? So now, if we talk about the gene in eukaryotes and prokaryotes, if we compare the gene in eukaryote and prokaryote, so genes in eukaryote and genes in prokaryote. So prokaryotic genes, they are very simple. Prokaryotic genes, how they are? They are very simple. So the gene will start at a point and it will end at another point, and all the sequence in within or all the nucleotides in between, they will code for protein. Okay, so they consist of continuous coding regions. They consist of continuous coding regions, prokaryotic genes mostly. Okay, but in case of eukaryotic gene, if we discuss, eukaryotic gene means coding sequences in eukaryotes. See, there is coding and non-coding, right? Coding and non-coding. But if we take some of the coding sequences, in that also, in the coding region as well, which is going to give rise a complete protein, that also intervened by some non-coding regions, some non-coding regions, means some of the sequence is coding for amino acid, again the sum is not, right? Again some of the sequence is coding for amino acid and some of this not. So this particular coding and non-coding region within a gene, right, I'm saying within a gene, right, this complete is a gene, but within gene also, in case of eukaryote, there are some coding and non-coding regions. So coding regions in within the gene is called as exon. Is called as what? Exon, the expressing region. Exon. And non-coding region is called as intron, which is inert. Right? Just a minute, give me a second. Am I audible? Am I audible?
Yes, ma'am.
Yes. So introns and exons are there. So you can remember exon EE. You can remember EE. Exons are the expressible region. So in case of eukaryote, these all the expressible regions, they are joined to each other. Okay. So first what happens? First, the gene is being completely synthesized into the RNA. So the gene is completely synthesized into the RNA. Then the coding RNAs, they are spliced together. What is done? The coding RNA part, they are spliced together. So these sequences, they are spliced together, and these exons, they are spliced together, and these introns, they are removed. Okay, so this is called as removal of introns, and after that, now these will encode the proteins, right? So see now here, can from this, so now I want the answer that we stated that from the amino acid sequence, right, if we know the amino acid, number of amino acid, we can give the length of DNA in the length of gene in the DNA, right? If we know the amino acid, the number of amino acid, we can give the length of gene in DNA. So will now this length will be correct? Will now this length of gene will be correct? Getting what I'm trying to ask, students? No, right? So when the gene is consist of coding and non-coding regions, that is exons and introns, that time we can give the minimum length of gene, right? कम से कम इतना length तो होगा ही, but if there is some intervening introns are there, the length of the gene can increase the actual length, right? So see what we stated, with the information of number of amino acid in the protein, we can give the length of gene, or we can give at least minimum length of gene. Understood? Clear? Yes? Clear everybody? What happened? So now let us discuss about the eukaryotic gene packaging. Now we get to know how the prokaryotic DNA is. It is mostly single chromosome. Most of the prokaryotes, they have single chromosomes and circular DNA. The eukaryotic chromosomes, they are mostly kept in fragments, right? One fragment is equal to one chromosome, and in the genes of eukaryotes there is exons and introns, right? Now let us talk about the packaging of this DNA. So the packaging of DNA leads to formation of chromatin. Packaging of DNA leads to formation of chromatin and the DNA packaging. Now we are talking about the DNA packaging in case of eukaryotes. Okay, we can take the example of plants, animals, okay, like humans. So this is done with the help of supercoiling. So packaging of DNA is done by supercoiling. So we need to first understand what is supercoiling. So to understand best what is supercoiling, here we have the example of the phone wire, right? So we can say here the wire of the phone, landline phone, is already coiled, right? Is already coiled, है ना? Like our DNA, the two strands of the DNA, they are coiled within. So this is called as coiling, right? This is a coil. But if we are coiling this coil on one another like this, if we are coiling this coil with one another like this is called as super coiling, right? So to pack DNA, we need to do super coiling, right? So how we are going to supercoil the DNA? So yesterday we have introduced some proteins, right? So what is the supercoiling of DNA? Supercoiling of DNA can be looked like this. So circular DNA can be supercoiled and the linear DNA can also be supercoiled. So the double helical structure is a coil structure, right? And when we are interwinding this double helical structure, so that is a supercoiled structure. Okay, understood? So when the axis of the DNA double helix is coiled on itself, so double helical axial, when we are coiling on itself, so that is becoming a super helix. That is called as supercoiling, right? So first we need to understand what is to understand this, we need to understand two concepts, that is underwound DNA and overwound DNA. Okay, so whenever there is DNA, what is underwound DNA? So when the DNA is under unwinding, okay, it is already wounded, it is already supercoiled, and when it is required for any process like replication and transcription, so we are unwinding it. So already wound DNA is called as underwound DNA, and when we are opening it and rewinding it, है ना? So that is called as overwound DNA. Okay, yes, getting everyone? So this coiling of the DNA is done by some enzymes, with the help of some enzymes. So now we are going to talk about that enzymes which are going to help us to coil the DNA. So coiling of the DNA is done by the topoisomerase. What is the name of enzyme? Topoisomerase. So, as we have discussed yesterday, the supercoiled structure of the DNA, that is chromosome structure or chromatin structure, is formed by wounding the DNA around some proteins, right? So, this structure is made by some with the help of some protein. So, protein plus DNA is nothing is equal to chromatin, and the compaction, the supercoiling of chromatin gives us chromosomes, right? So yesterday we introduced name of this protein, so these are called as histone proteins, right? So these histone proteins are of 4 type as we discussed: H1, H2A, H2B, H3 and H4, right? H1, H2A, H2B, H3 and H4. These are 5 histone proteins. So among these 5 histone protein, the 4 H2A, H2B, H3 and H4, the 2 units of each, you can see the structure, the 2 units of H2B, 2 units of H2A, 2 units of H3 and 2 units of H4, they combine together to give rise a bead-like structure called histone octamer. This is called as what? Histone octamer. And on this histone octamer, the coiled DNA is supercoiled, right? The coiled DNA is wounded actually, right? This is coiled DNA. The helical DNA is called as coiled DNA. So see, now let us talk the stages in the coiling of DNA. So this is your helical DNA, or we can say coil DNA. So the diameter of this DNA is 2 nanometer. What is the diameter of the DNA? 2 nanometer. So when these DNAs, when they are wounded around the histone octamer and making a bead on string structure, this is called as what? Sorry, string on bead. String on bead structure. It's looking like the string is wounded around beads. So that is called as nucleosomes. Here is the chances of question being asked. What kind of question can be asked here? That the sequence of the packaging of DNA. Okay, what is the sequence of packaging of DNA? So first there is DNA, then there is nucleosomes. These nucleosomes give rise to solenoid model, and then it is extended to form a chromosome. Okay, so DNA, nucleosome and chromosome, this is the sequence. So see, when the DNA, when it is bonded on the beads, its nucleosome, now the diameter of this DNA increases because it is now more compact. So it is now 11 nanometer. Now one thing we need to understand here, so whenever there is bounding of the DNA on this on these beads, when we are wounding the DNA, the length of DNA we are wounding over one bead is 200 base pair. Okay, or the length of the DNA from one bead to another bead is 200 base pair. So how many base pair they are wounded around? One bead is 200, but among this 200 base pair, 149 base pairs they are on bead. 149 base pair they are on the bead, and the remaining base pair they are linking one bead to another bead. The remaining base pair they are linking one bead to another bead. So you can see here. So here on this bead there are 149 base pair, and the remaining are the linking one, that is how much? 51 base pairs, right? So this complete is 200 base pair. So the DNA which is in between two beads of histone is called as linker DNA. Is called as what? Linker DNA. And who is helping this linker DNA to withstand is H1 protein, right? We have seen that there are 5 types of histone protein, right? H1, H2A, H2B, H3 and H4. So among these 5, these 4, they are being involved in the formation of histone octamer. So where is H1? So H1 is actually here, which is acting as a linker histone. So H1 is called as linker histone, and the sequence to this linker histone is called as linker DNA, which is linking the beads of these histone proteins with the DNA. So there is linker DNA and linker protein. The length of the linker DNA is 51 base pair and the linker protein is H1. And the complete length in one bead of a nucleosome is 200 base pair. So this structure formed is called as nucleosome. So now these nucleosomes, they get compactly packed with each other to giving rise of 30 nanometer fiber. So this 30 nanometer fiber is also called as solenoid fiber, or this is also called as solenoid model. This is also called as what? Solenoid model or solenoid fiber. Getting everyone? Yes. So the further packaging of DNA. Yes, may I have some responses? Do everyone getting? Yes. Okay. So now here try to understand. Yes. Okay. So here try to understand from nucleosome. From nucleosome, further we are going to give rise the solenoid model, right? So a 30 nanometer fiber, right, which is higher order organization of nucleosome. So we can say condensed chromosome structure, right? And to make condensed chromosome structure, first we have converted DNA into nucleosome, and then this nucleosome into the a thick structure, right? Called the nucleosomes. They are coarse, right? Seems to be organized into the 30 nanometer fiber, right? Which is called as. So now this conversion of DNA into 30 nanometer uh, rod like structure fiber can be done in two type. It can be done in two type. So first is, so this 30 nanometer fiber can be obtained in two type. One is one is solenoid fiber, one is solenoid fiber, and another one is zigzag fiber. Another one is zigzag fiber. So if this nucleosome, it is it is twisted left-handedly. If the nucleosome is twisted left-handedly, one start helix is there only. Okay, there is one start helix and it is twisted left-handedly. See, like this. If only one start helix is this and it is twisted left-handedly. So this is called as solenoid model. Okay, and here in zigzag fiber there is two start of helix. Two start of helix. So there will be two nucleosomes like this, and they are again interwounded. So there is two start of helix, right? There is only one start of helix. So this 30 nanometer fiber is called as zigzag fiber, and if it is only one start of helix and left-handedly twisted, so it is called as what? It is called as solenoid fiber or solenoid model. Most of the organism they follow the solenoid model. So now this nucleosome is now it is 30 nanometer, right? First it was 11 nanometer nucleosome, now that is 30 nanometer nucleosome fiber, which is called as also solenoid model. Now this solenoid model is extended. It is compacted to give rise a extended form of chromosome, right? It is not now exactly chromosome, it's an extended form of chromosome of 300 nanometer. Now this is again condensed, compacted to give rise a diameter of 700 nanometer, more compacted form, right? And finally, so this is the final chromosome, condensed part of chromosome. Now if this chromosome, it will get duplicated in process of cell division, so it will be 1400 nanometer, double of the 700 nanometer, right? Because in case of cell division, the chromosome will get duplicated, right? Understood everyone? So what is the diameter of a compact chromosome? 700 nanometer. What it is? 700 nanometer, right? So which enzyme is so much important to do all this supercoiling? Is the topoisomerase. You have to remember the name of this enzyme, topoisomerase. More properly it is topoisomerase 2. More properly it is topoisomerase 2. Got this much clear? Yes?
Clear.
Okay, great. So now one more thing we need to understand here is there is coiling of DNA, right? Coiling of DNA lead to the formation of chromosome. Now sometimes, so when we are de uh, so in the coiling of the DNA and making the chromosome, right? So we have made the chromosome. When this chromosome is going to duplicate in the cell division, right? So in case of eukaryote, see we are talking about the eukaryotic chromosome, right? Packaging of the eukaryotic DNA. So we get to know that the DNA to nucleosome to the solenoid fiber of 30 nanometer. Then there is the fiber of 300 nanometer and then 700 nanometer complete chromosome. If we duplicate it, it will become the 1400 nanometer duplicated chromosome. So to keep these chromosomes aligned, there are two protein important playing role, that is cohesins and condensins. Cohesins and condensins. Okay, so cohesins and condensins, they are very important in cell cycle. They are very important in the cell cycle of eukaryotes. So we have chromosomes, right? Now we have chromosomes and we are going to talk about the two important structures in the chromosome, that is centromere and telomere. Okay. So now these chromosomes we have to duplicate, right? These chromosomes we have to duplicate, keep them together, and when there is time of division we have to again separate them and send into the two different type. So to maintain them, to keep them together, to separate them, two proteins they are helping us, that is cohesin and condensin. For now we can remember this much only, that cohesins and condensins, they help to maintain the chromosomal structure during the cell division. Okay, so these cohesins, they help in whenever the cell chromosome get duplicated, right? Am I audible?
Yes, ma'am.
Hello. Okay, so see here, the role of cohesins and condensins, right? So when these chromosomes they are getting duplicated, right? So these chromosomes, this is a single chromosome, right? When it get duplicated, the now this is called as what? This the part of the chromosomes, two halves of the chromosomes, they are called as chromatids. They are called as what? Chromatids. Okay. Chromatin is different, chromatid is different, right? Chromatin we learnt the compact form, right? That is chromatin, which is formed from the. So where exactly we called chromatin? Nucleosomes. When we pack nucleosome, it is chromatin. Okay. So chromatin they are made up of nucleosomes. Now these are what? These are chromatids. So whenever there is duplication of chromosome, there are two chromatids, right? Duplication of chromosome leads to formation of two chromatids. Now these two chromatids they are called as sister chromatids. What they are called as? Sister chromatid. So this cohesin proteins, it helps in keeping together the sister chromatids. Okay, understood? And condensin will help in compacting and again separating the protein, cohesin and condensin. Both of them. Hope this much clear. So now let us talk about the two important parts on the chromosome, that is telomere and centromere. So now we are going to discuss about the telomeres and centromeres. So can anybody tell me what is telomere, what is centromere? Yes?
Telomere refers to end of the chromosomes.
Okay, telomere refers to the end of the chromosome, and centromere?
It's like center.
Center. Okay. Chalo. S- Nice. ठीक है. So up to now we have learned that after compacting the chromosomes, they will look somewhat like this, right? So centromere are the constriction on the chromosomes, right? They are the some regions of the chromosome, or we say constrictions on the chromosomes, which are mostly in the central. So the centromeres is a sequence of DNA that functions during cell division as an attachment point for protein that link the chromosome to mitotic spindle. What is centromere? So centromere is the constriction, we say. Okay, towards the central. Centromere is the primary constriction on chromosome, right? So, very good. So, what is centromere? Centromere is the, right, we say primary constriction on chromosome, which is what? Which is actually the point. It's a constriction. It's a constructed part, constructed in a such a manner that it's going to be a point where the spindle fibers can attach. Spindle fibers can attach, where the spindle fibers can attach. So see, at the time of cell division, chromosomes, they are somewhat like this, and they has to be separated in two halves. So to separate them, some pulling fibers, they are attached to this chromosomes at the point where they are going to attach, that is called as centromere. And over the centromere there is a protein called kinetochore. Okay, centromere is the part of DNA actually, right? So over the centromere there is one more protein called kinetochore, and actually the spindles, they are going to attach to the kinetochore, right? And spindle fibers, they arose from where? They arose from, yes,
Microtubules.
micro. Okay, spindle fibers are also called as microtubules, but they arose from which structure? Yes, spindle fibers are also called as microtubules. Suppose this is a cell, right? So here is some structure called like this, है ना? Books में वो ऐसे draw किया हुआ रहता है ना? होता है क्या? Like this, right? So from this, actually, those microtubules, we say spindle fibers, they arose, right? And they attach to the centromere of the chromosomes, right? So I am asking what this is called as, from where the centromeres arose. Sorry, from which the spindle fiber arose. What this structure called as? It is called as centrioles. It is called as what? Centrioles. So see, so many similar words. Chromatin, right? Chromatids, centromere, then centrioles. Each of them has different function and they are different from each other, right? Chromatin is what? Next step to the nucleosomes, right? Chromatid is the arms of the chromosome. Centromere is the primary constriction, and the centriole is the site or the structure from which these spindle fibers are going to arise, right? And the point where the spindle fibers are going to attach, it is centromere, right? Yes, clear everybody? Clear about centrioles, centromeres, right? So centromeres, right? The attachment which is essential for the equal orderly distribution of chromosome sets in the daughter cells, right? That's why the centromeres, they are important, right? So mostly the sequence in the region of centromere, if we, so there are so many studies. So it says that mostly this centromere region, in case of Saccharomyces cerevisiae, in case of yeast, it is made up of 130 base pair. Is made up of what? 130 base pair, and the centromere region, the length of the centromere region is 130 base pair, and it is very rich in ATGC sequences. Which sequences? ATGC sequences. Understood? Yes. Now let us talk about the telomere. Yes, now we'll discuss about the telomere in the chromosome. Now we know how chromosome is formed. So we are now talking about the parts of chromosome. So now let's discuss about telomeres. So what is telomere? Sequence at the end of eukaryotic chromosome that helps to stabilize the chromosome. What is telomere? Sequence at the end of the, see, these telomers, they are not present in prokaryotes. Eukaryotic chromosomes, which helps to stabilize the chromosome. Okay, so the telomere are the sequences which are consist of repeating units, or telomere is the repetitive sequence. How telomere is? It's a repetitive sequence with non-coding region. Telomere is repetitive sequence with non-coding region. Why it is repeated, and the why the sequence is non-coding there? Can anybody tell me? Yes? Yes? Anybody? So see, if this is your chromosome, if this is your chromosome, this will be your centromere, right? And sequences at the end, these are nothing but the telomeres. So why eukaryotes has telomeres? Why there is need of keeping such repetitive sequences which are non-coding? You know what? It's to manage end DNA replication error. It's to manage what? And DNA replication error. Okay. You know what? As we get older, as the age of an organism increases, the telomere, they are going to decrease. As the age of telomere, as the age of organism increases, telomeres, they are going to decrease. So telomere shortening, right? Telomere shortening is one of the, I am saying one of the, okay, is one of the reason for aging. कोई बताएगा क्यों ऐसे रिपीटेटिव सीक्वेंसेस हमने रखे हैं and why these sequences they are getting shortened and why they are so much important for us. Yes? Anybody? No तो no बोल दो. So I will tell you. Yes? No? Okay. So let's discuss. It's a very interesting topic. See, it's related to aging. So how the human DNA is? How the human DNA is? It is linear. How the human DNA is? Linear, or most of the animal DNA is linear. We talked about in case of prokaryote, it's circular, right? So when the replication of the circular, we are going to talk in replication, this topic, okay? We're going to discuss on this on the replication. So when the replication in the prokaryote it start, so it is bidirectional. In both the direction the replication starts and it stops at one point, right? So the complete DNA is being synthesized. But in case of eukaryotes, the DNA is how the DNA is linear, and this is also going to be bidirectional. The replication of the DNA is also going to be bidirectional. So let us draw the linear DNA, and in between we are going to start its replication. So suppose this is a point where we are going to start the replication. So to replication to happen, we are going to unwind the DNA. We are going to open up the DNA, right? So this is an open part of the DNA, right? So from this, the replication start in the both the direction, right? If the replication is started in this direction, the second strand will be synthesized in this direction, both the strands. See, when we are saying replication is bidirectional, so at the point where replication starts, there is a synthesis of four strands. Okay, iske do strand or iske do, right? Again, if it is in this direction, if it is synthesizing in this direction, it is going to be synthesized in this direction, right? We learned yesterday that the direction of the synthesis of DNA replication is 5 prime to 3 prime direction, and two strands they are opposite to each other, right? They are complementary to each other. So their uh, replication, the direction of the replication is going to be opposite. So when the replication starts, it's go to the end, right? The replication is going to end. So at the end, there is slippage error. The polymerase, the enzyme which replicates the DNA is called as DNA polymerase. So at the end sequences, this enzyme DNA polymerase, it slips from the end because these ends are open ends, right? So it's going to slip at the ends. So the ends of the DNA, they are not completely synthesized with each and every cycle. So the ends of this DNA, they are getting shortened and shortened because of the end replication error in case of the linear genome having organisms, right? In case of the chromosome. So this is an end DNA replication. So if suppose at the end there are some genes, will they be going to code? No. Will they be going to pass into the next cell? No. So at the end of the DNA, there is kept non-coding repetitive sequences, and at with each cell cycle, this telomere is going to shorten and shorten. But at one stage the sequence of the telomere is finished, right? Now the important sequence of this DNA is going to what is going to lose, right? We are going to lose the important sequence of DNA, and this leads to the loss of some functional proteins from the cell, leads to aging. Understood? So actually the length of telomere also decides the age of an individual. Getting? Yes? Yes or no? So this is the importance of telomeres in the eukaryotic DNA, right? So they minimizes the end replication error. Understood? Clear? So this is all about the chromatin. What is chromatin? Chromatin structure, the DNA organization in eukaryotes and DNA organization in prokaryote. So the DNA in the prokaryote is also compactly packed instead of, so this circular DNA is going to supercoil, right? It is already double stranded circular DNA, and that is going to circular supercoiling will be there in case of prokaryotes, right? And in how the supercoiling is in eukaryote, we have already talked about it, right? And the enzyme, important enzyme helping us to do so is topoisomerase. Most importantly, it is topoisomerase 2, right? Clear? So now I have one question for you all that you have to think on it, okay? So if the telomere is being shortened, right? If the telomere is being shortened with each and every cell cycle, with each and every cell division, so how? So age, so complete complete, if we completely exhaust the telomere, it leads to aging, right? Now the individual is old, है ना? Actually it is making the individual old and old, but this individual is going to pass the gene, or we say the genome DNA, to the next organism, right? To the next generation. Then also, even though the telomere of this individual is getting shortened and this individual is reproducing and giving rise to the new individual, then also the average lifespan of both the individual is same. How come? Ideally it should decrease, right? Getting the question? The average lifespan of all the individuals of a species is same. So even though there is telomere shortening, how the average lifespan of all the individual is same? ऐसा है तो ऐसा होना चाहिए ना कि जो फर्स्ट जनरेशन है उनकी लाइफ स्पैन ज्यादा होनी चाहिए, नेक्स्ट जनरेशन वालों की लाइफ स्पैन कम, और उसके नेक्स्ट जनरेशन की और कम होनी चाहिए, राइट? यस और नो? एम आई आस्किंग राइट क्वेश्चन और नॉट? Yes. If I'm saying that telomere shortening leads to the aging, right? So see, if you say all the individuals, right? If we talk about humans, so mostly the reproductive age of the individual is after 18, right? Or after 16, 15 to 16 year, they get reproductively able, right? Mature. Puberty is there. So till then, there are many multiple cell divisions done in the individual, right? So telomere is already shortened, and at that time, if they are producing gamete, so how this gamete has the equal age as the parent, average equal, right? समझ में आ रहा है question, है ना? Isn't it interesting? Yes. So today we have complete all the topics up to we have planned. So from tomorrow we are going to talk about the next topics. Okay, further topics we're going to talk about. Or today you have time to revise and search and think upon the question I have asked to you, right? है ना? Everyone can think on this, right? If you got the answer, if you researched, you can get back to me and tell me. So from tomorrow, we are going to talk about the molecular genetics. Okay. Yes. So first we will talk upon as we have learned about the DNA. So we will continue with the replication of the DNA, how we can duplicate this DNA, right? And we know that this DNA contains genes which is going to give rise us the product, gene product, like it can give us RNA and protein. So इसी topic को continue करते हुए we'll talk about the a replication, transcription, translation, and then further we will talk about the principle of inheritance. Okay, clear? So everyone is going to revise and think over the question I have asked. Yes?
Ma'am,
Yes?
ma'am, is it the relation between germ cells and somatic cells? The question you asked.
Yes, yes, yes. You are coming close. Okay. Yes, germ cells and somatic cells. The individual it passes germ cells to the next generation, right? है ना? Okay. So tomorrow we are going to discuss what special this germ cells contains, है ना? That making them able to give the next generation the complete DNA. Okay. Yes. Okay. So for today we are going to stop here, and let's discuss the further topics in the next. Bye everyone, take care.